Comparative genomic hybridization is a molecular cytogenetic method for analyzing copy number variations (CNVs) relative to ploidy level in the DNA of a test sample compared to a reference sample, without the need for culturing cells. The aim of this technique is to quickly and efficiently compare two genomic DNA samples arising from two sources, which are most often closely related, because it is suspected that they contain differences in terms of either gains or losses of either whole chromosomes or subchromosomal regions, a portion of a whole chromosome. This technique was originally developed for the evaluation of the differences between the chromosomal complements of solid tumor and normal tissue, and has an improved resolution of 5 to 10 megabases compared to the more traditional cytogenetic analysis techniques of GEM subbanding and fluorescence in situ hybridization fish, which are limited by the resolution of the microscope utilized. This is achieved through the use of competitive fluorescence in situ hybridization. In short, this involves the isolation of DNA from the two sources to be compared, most commonly a test and reference source. Independent labeling of each DNA sample with fluorophores, fluorescent molecules of different colors, usually red and green. Denaturation of the DNA so that it is single-stranded, and the hybridization of the two resultant samples in 11 ratio to a normal metaphase spread of chromosomes, to which the labeled DNA samples will bind at their locus of origin. Using a fluorescence microscope and computer software, the differentially colored fluorescent signals are then compared along the length of each chromosome for identification of chromosomal differences between the two sources. A higher intensity of the test sample color in a specific region of a chromosome indicates the gain of material of that region in the corresponding source sample, while a higher intensity of the reference sample color indicates the loss of material in the test sample in that specific region. A neutral color yellow when the fluorophore labels are red and green indicates no difference between the two samples in that location. CGH is only able to detect unbalanced chromosomal abnormalities. This is because balanced chromosomal abnormalities such as reciprocal translocations, inversions, or ring chromosomes do not affect copy number, which is what is detected by CGH technologies. CGH does, however, allow for the exploration of all 46 human chromosomes in single test and the discovery of deletions and duplications, even on the microscopic scale, which may lead to the identification of candidate genes to be further explored by other cytological techniques. Through the use of DNA microarrays in conjunction with CGH techniques, the more specific form of array acgh has been developed, allowing for a locus-by-locus measure of CNV with increased resolution as low as 100 kilobases. This improved technique allows for the etiology of known and unknown conditions to be discovered. Subscribe our channel for more such informative short video.